We always hear about DNA as the blueprint of life, right? But what if I told you that blueprint, well, it doesn't really tell the whole story. Today, we're going to dive into the world of proteomics, a field that looks past the instructions to see what's actually happening inside us minute by minute.
So, we all learned in school, right? DNA makes RNA, and RNA makes proteins. It's the simple, elegant flow of information. But think about it this way. What happens when the blueprint calls for 100 workers, but only 10 actually show up for the job? Or worse, what if they start doing tasks they were never even assigned? See? That's where the blueprint starts to fall short.
This analogy, man, it really hits the nail on the head. A city blueprint is absolutely essential, but it won't tell you about a traffic jam on the freeway or a sudden power outage or a surprise street festival. To understand how that city is actually living and breathing, you need to watch the workers, the cars, the whole shebang. That's the proteome. It's the complete set of proteins that are out there right now getting the work done.
Okay, so let's really dig into what makes proteomics such a game changer and why it takes us so much further than what we can learn from studying the genome alone.
So, to put another way, if genomics is like a giant cookbook that lists every possible recipe a chef could ever make, proteomics is like walking into the kitchen and tasting the actual dish that's on the stove right now. It tells us exactly which ingredients were used, how much of each, and how it all came together in this one specific moment.
Now, I know what you're probably thinking. Hold on. If the DNA has the code for all the proteins, why can't we just read the DNA and call it a day? And you know what? That's a fantastic question.
And the answer is that an unbelievable amount of editing, tweaking, and flat-out regulating happens after those initial instructions are read from the gene. The biological reality is just way more messy and way more interesting.
Yeah, the journey from a gene to a protein is less like a simple assembly line and a lot more like a chaotic gourmet kitchen. The initial recipe from the DNA gets all sorts of last-minute changes. It gets spliced, edited, and folded into crazy shapes. The final protein might even get things added to it or have bits chopped off after it's been cooked. And the result of all this? A single gene can end up producing dozens of different proteins, each one with its own unique job to do.
And this right here is the key takeaway. The proteome is alive. It's constantly changing. It's reacting in real time to everything, from the lunch you just ate to a virus that's trying to invade. So if you really want to know what a cell is doing, if it's healthy or if it's sick, you can't just stare at the static blueprint. You have to go down to the construction site and watch the workers themselves.
Okay, this all sounds incredibly complicated, doesn't it? So how in the world do scientists take a tiny sample with thousands of different proteins all mixed up and figure out what's what?
Well, the answer is this amazing machine called a mass spectrometer. At its heart, it's basically a hyper-accurate molecular scale. I mean, it can weigh individual molecules with such insane precision that we can figure out exactly what they are just based on their mass. It's incredible.
And here's the cool thing. No matter how big or complex they look, every single mass spectrometer has the same 3 core parts. There's a source to get the sample ready, an analyzer to sort everything, and a detector to count the results. It's a simple 3-step process designed to make sense of molecular chaos.
The journey a protein takes through this machine is a pretty wild ride. It's a mix of some crazy physics and chemistry, but it all boils down to these 3 essential steps.
First up is ionization. See, you can't really control neutral molecules with electric or magnetic fields. So the very first thing you have to do is give them an electric charge. You've got to turn them into ions.
And there are a couple of really clever ways to do this. One method, called MALDI, uses a laser to gently zap the proteins and launch them into the air. Another one, called ESI, uses high voltage to turn a liquid sample into a fine, charged mist. But for both, the end goal is exactly the same. Get those proteins charged up and flying through a vacuum.
Okay, so our proteins are charged up and flying. Now they enter stage 2, the mass analyzer. And honestly, the absolute best way to think of this part is as a racetrack for molecules.
One of the most popular types of analyzers is called time of flight, or TOF for short. Here's how it works. All the ions get the exact same push of energy. But, just like in a real race, the lighter ones take off and accelerate way faster than the heavy ones. So by timing exactly how long it takes each ion to fly down this flight tube and reach the end, we can calculate its mass with incredible accuracy.
The final step is detection, and it's pretty straightforward. At the very end of that molecular racetrack, there's a detector that acts like a finish line. It just sits there and counts each ion as it smacks into it.
and it records precisely when it arrived.
So what you get at the end of all this is something called a mass spectrum. It's a graph that shows every single molecule that was detected and how much of it there was. Each peak on the graph represents a specific molecule, creating a totally unique chemical fingerprint that we can now use to identify every single thing in our original sample.
So, we've got these incredibly detailed, specific fingerprints of all the proteins in a sample. What's the payoff? What can we actually do with all this information?
Well, that chemical fingerprint isn't much use on its own. The next step is all computational. You can honestly think of it like Shazam, but for molecules. Scientists use these really powerful algorithms to compare their measured fingerprint against enormous databases of all known proteins, and they get a positive ID on everything that was in their sample.
And the impact of this is it's huge. By figuring out not just which proteins are there, but in what amounts, we can do things like diagnose diseases much earlier, discover brand new targets for drugs, and just understand the basic mechanics of life in ways we never, ever could before.
At the end of the day, proteomics isn't just about making a big list of parts. It's about taking a snapshot of the living machine while it's running. It's a dynamic, real-time picture that tells us what a biological system is actually doing right now in this very second.
And that leaves us with a truly exciting question for the future, doesn't it? As this technology gets faster, cheaper, and more accessible, just imagine being able to read a person's proteome in real time. What early warning signs of disease could we catch? And what could we potentially stop before it even has a chance to start?